Every few months, we all get a new upgraded mobile phone launched in the market. Isn't that right? Yes. But the same way, have we ever tried to upgrade ourselves, our skill sets, and our CV every few months? The answer for most students for this question will be no. And that is where I compliment all of you, all the students who, like you, who become successful because you never fail to upgrade on your skill sets. You never fail to attend Biotechnica's webinar. Now, on your path to success in your career, it is important to build your credibility and value. And this only comes from hard work and commitment. You have to show commitment and hard work in your field of work. Successful people never stop learning. They are always on a lookout for new trends, new skill sets, which they can acquire, which can help them create more opportunities for themselves. So, ladies and gentlemen, if you want to make opportunities for yourself, listen on. Hello, everyone. This is Shekhar Suman, CEO and Managing Director of Biotechnica and Rasainika. And today, as I come live from Bengaluru, welcome to the webinar on CADD Masterclass. Let's talk about this one trend that has changed the biotech and pharma industry's face. And this one skill which can propel your career to greater heights. Yes, I'm talking about computer-aided drug design. Now, we are well aware of how elaborate, how tedious, how time-consuming, how energy-consuming the drug discovery process is. Imagine if we were still dependent on the old technologies, old techniques, old methodologies. Would we be able to develop a vaccine, manufacture drugs at this rate in this pandemic era? The simple answer is no. It was not possible. In fact, a few years ago, if someone had suggested us that we will be able to develop a vaccine in a span of a few months, people would have laughed at them. People would have mocked at that person. But today, hats off to the scientific community and the scientists who are working tirelessly and the advanced technology they are using. The experts are now optimistic that we will have a 100% efficient vaccine for COVID-19 as early as January 2021. Now, all this is possible because of infinite amount of research and data available. And of course, technology to analyze and interpret it. Now, you as future scientists have to know all this, that when we combine the wet lab techniques and CADD, we can better optimize the drug discovery process. It has made the screening process quicker. It has cut down the cost of designing and much more. So let us understand what is CADD. Does it sound interesting to all of you? Please reply in chat. This is the future of life science. Yes, the future of life science goes hand in hand with technology, and this is one of them. So this is the right time for you all to equip yourself with all the knowledge available about CADD, computer-aided drug design. So if you are ready to learn, then type in the chat box, eager to learn, ETL. And then we will start our exciting webinar for all of you today, where our CADD experts are going to share some very interesting insights for all of you. Learn how to master this technology and improve your career options. As the British author Ken Robinson once said, curiosity is the engine of achievement. Stay curious. Always develop a passion for learning. And if you do so, you will never cease to grow. And that's one message I wish to give you today. All the best for your career, and I will meet you at the end of this webinar. Thank you so much for listening in, and I'll pass on the controls to our team of experts. Till then, take care. Bye-bye.
Thank you, Shekhar sir, for the wonderful introduction. Hello everyone. Please confirm if the slide is visible to you and I am perfectly audible to you. It's loud and clear enough. Yeah? Okay. Fine. Thank you. Now today's session already introduced by sir, that is computer aided drug designing. The question is, yes, we do need drugs in our everyday life, right? In the sense to treat diseases or to cure any condition. Even right now, the one question that everyone is having is, when will the treatment for COVID-19 come, right? Now, how we can design, if you look at the traditional process of drug designing, which was done in historical times, it was to identify a certain molecule which can go and bind to a target, or can go and inhibit an enzyme, or can activate the gene expression of a certain protein. All these are related to a diseased condition. In case something is not working fine, then what if we can include some molecules which can correct that particular faulty condition. That was about the concept of drug designing, but that is only, I could explain it in 1 minute, but if you look at the actual procedure, it's really very long. Now today, these newer times when we have artificial intelligence with us for our service, so why not use it? Why not make this complex process a lot more simpler? And for that only we require the computer aided drug designing, some knowledge about it. Now let's say we have a mixed group of audience over here, beginners and experts or intermediates as well, but this is just an introductory session which will be giving you the idea and the outline of what exactly is done in this process, right? Now, to understand what exactly we are doing, I'll ask you some question. How many of you have bought spectacles or sunglasses online, which you can try it on your face, right? So what do? We have a particular conformation of the face, right? And on that conformation, what particular molecule, that is a sunglass, it will suit, it will look good, right? So here it's just looking good, but when it comes to actual drug, that is which will be able to go and bind properly, right? Which will be fitting properly. Or even if you see, you have witnessed some home interior designing, what they'll do is a virtual simulation. In the sense, they will have some, there also it is computer aided, they'll have some software or some application or some tools, and they will take the measurement of your house and they will input in the system, and you will have a virtual simulator of the house, right? And then you want to put a cupboard over there, or a sofa over there, or a bed over there, and then you have particular conformational space, and in that space what will come and fit properly. Obviously you don't want a couch so huge that it will oversize your entire drawing room area, right? So, or else you don't want so small that it will be very insignificant. So that's why you need a perfect thing. And also, if you have a wall, L-shaped wall, then you need a sofa which is also L-shaped, just to match with the wall, to fit properly and confirm, align properly with the wall, isn't it? Or even a simple thing, when we go out shopping, right, in the mall or somewhere, now what do? We will try out the different clothes which is fitting us properly. The fit is what we first see before buying any clothes, isn't it? So why not use this simple example when we think of a particular protein and we think of a particular target, whether the target protein, what kind of molecules will be able to go and bind to them, because everything that is happening, any disease conditions or any symptoms, those are all the play of molecules and their pathways and processing and signaling, gene expression. So molecules control molecules. That's what the basis of the drug designing process, right? Now when we were talking about traditional process of drug discovery and development, the computational approaches in drug design, then discovery and development, they are gaining very rapid exploration, implementation and also admiration. Now when you have to introduce a new drug in the market, it is a very complex, risky and most importantly it is a costly process. So in terms of money also, in terms of time and even manpower. Generally it is found that drug discovery and development process takes around 10 to 14 years and more than $1 billion capitals in total. Now if you are selecting, obviously directly you cannot jump off to the drug, right? When I'm talking about identifying some molecule which has the potential of forming a drug, you cannot find it just like that. You will have so many choices. From there you have to identify it. So there, from the starting compounds will be a whole lot, obviously even more than that. And then this entire jumping off to the preclinical phase, this preclinical and clinical, they are just the trying out whether it is actually working or not. But before that you have to develop it, you have to identify the molecule, optimize its characteristic. Out of that you have to select which will not give toxicity, which will not give adverse effects. Many such concepts you have to have a check for that, okay? And then only you can have some, out of that many you just have 250 compounds. And preclinical phase, when it actually moves to the human trials, the clinical trials, which is a long phase, again you will see that half of, like more than 90% of the molecules will get rejected. So only 5 of them probably will enter the clinical phase, and out of that, if everything is going fine, there are no adverse effects or side effects, then only the drug will be approved, right? So for reducing time and cost and risk-borne factors, computer-aided drug design method is widely used as the new drug design approach, because at least we can reduce in this phase. See, clinical trial we have to do, but even now they are taking the history of the patient and they are trying to analyze what can the drug do in the body. All that is okay, but at least this part they can make it come down to a lot shorter time frame, okay? Otherwise if you have to do it experimentally, then it will take a lot of time. So it has been seen that by the use of CADD approaches, we can reduce the cost of drug discovery and development up to 50%. And CADD consists use of software program based processes, so that we can establish a standard to relate activity to structure. So we need some standardization of, if this particular structure is there for the molecule, what kind of activity it is going to give, right? How many of you have worked with this drug design remotely, if you have been associated either in any industry or any company, even in the management sector or in any of these fields, right? So this hardcore field, obviously mainly the principal investigators will be investigating their projects like that, but at least how many of you have been involved in this process? So I'm sure you would agree, all of you, don't you agree, all those who have worked, that this process is extremely tedious, isn't it? So in fact in this COVID era also, it's been almost 1 year and we still don't have yet a proper kind of a treatment, right? So this is, anyhow 1 year is too less for a drug to develop, isn't it? So when the disease is understood at the molecular level and the target molecule are defined, drugs can be designed specifically to interact with the target molecule in such a way as to disrupt the disease. Yes, that's what exactly we will be doing. We have to understand the disease and we need to understand which protein molecule is responsible for that disease, whether it is getting overexpressed, whether it is not able to bind to its target. Many things we have to analyze. So that literature review we have to do before we begin our drug designing process. So accordingly, whatever is our target protein, accordingly our drug can be designed specific and complementary to that particular target, so that it can interact in such a way that the target will then behave in the way it is supposed to behave in the body, isn't it? Now the selected or the designed molecule, first thing is it should be able to interact with the target and it should be able to bind to the target, isn't it? Moreover, there are some other requisites. It should be a organic small molecule, it should be complementary in shape to the target, and thus it should be able to bind to the target and interact with the target, as I said. And it should be able to activate or inhibit the function of a biomolecule such as a protein or an enzyme, etc. Like as I said, the disease can be caused either by activation or inhibition of the protein, and you will require the reverse process to be done by your drug. So that's what the drug should be able to do. Now these are very basic steps what you need to do. You have to create your molecule, the druggable molecule, you have to identify that. Now sometimes what we do, we can identify the molecule from already existing molecules, alright, but sometimes all that you will learn it in later, like you will get an idea later. If we don't have a particular ligand, or we don't have the particular target structure known, then we may have to create the molecule. So that is also there, right? And then you have to dock the molecule. So docking means aligning, like in the sense like you have to see the fit, that's all. Like how you are docking the sunglasses over your face, the example I gave you, right? So dock the molecule to the target protein and see which are the binding sites, which are the complementary sites, where they are interacting with each other, right? So that's what you have to do, to analyze the molecular interactions. And not only that, you have to estimate what is the binding strength. So based on that, you will be getting some scores. So which drug will be having the maximum score? It will be done by on various factors. What groups or what functional groups they are binding to, or how much affinity they are having, and lot more such characteristics will be there. And then only you have to estimate their drug like property. So this is only the drug development part. Later the preclinical trial, if at all like it is selected now as a particular molecule, perfect molecule, that can enter into the preclinical trial. So before that also you have to do some optimization and toxicity testing, then only it will enter into the preclinical trial, right? So what exactly is CADD, computer aided drug design? So these are computational methods and resources that are used to facilitate the design and discovery of new therapeutic solutions for any conditions in your body. It involves the application of computational software and chemistry simulation. We can see the online simulator of the molecule, how it is binding, how what kind of homology sequence it will be having, right? And it will help identify any kind of novel hits or leads. So hits and leads in drug discovery terms, these are simply molecules. Now hits means out of that how many you have hit, like out of the entire range of molecules, how many are fit to be the particular drug of interest in the process which you are trying to cure. So out of those molecules, those are called as hits, which are fit for your further experimentation. Now out of those hits, obviously you are not going to select all these molecules. You have to select out some specific ones with specific characteristics. Okay, some will get rejected because of some possibility will be there. There are many possibility actually, because of which that molecule will not be able to carry forward. Okay, so in that case, whatever you're rejecting and whatever you're selecting out of those hits, that becomes the lead. Now it's your lead compound which will further proceed towards the clinical trial. But before that, again optimization and all should be there, which is called as lead optimization and all that. So that will be done against selected therapeutic targets, as well as supporting medicinal chemistry lead optimization programs. So CADD methods can increase the odds of identifying compounds with desirable characteristics, speed up the hit to lead process, and improve the chances of getting your compound past the hurdles of preclinical testing. So if this first part itself, it is very much efficient and specific. Out of the hits, if you are rejecting the molecules, even if what you remotely think that may hinder the binding, so those can be in a very technical way can be removed off. So definitely you are speeding up the hit to lead process. Okay, so definitely computational screening, it can help to discover the lead compound faster, right? Now what exactly is CADD doing? It is changing the random screening against disease assays. So you have identified few molecules in the traditional way if you talk, and then you are running assays on like whether it is having an effect on the disease treatment or not, or else you are taking some natural products or synthetic chemicals, right, which you might think will have the potential to treat based on some similarity of similar molecules, right? So that is changed to rational drug design and testing. So rational means you are starting from scratch, right? Like rationalizing the target, and based on that you will be designing the drug. It's not any arbitrary process, but a very technically defined process, right? So you're speeding up the screening process. Screening process is extremely important in drug discovery, because out of so many potential candidates, you need to select the ones which will have your characteristic. So more efficient screening, it should be focused and target directed. And de novo designing, so that is also target. De novo means from scratch. If anything is not available, the target protein structure, it may be possible you've identified the disease, but the structure you have not yet identified. Okay, so on what basis you will build the ligand or your drug molecule, right? Whether it's inhibitor or activator. So in that case, you have to start with ab initio, like some kind of de novo designing. So that will be the target directed, right? And integrating testing into design process, you can make the testing process also easier, and removing inefficient drug candidates which are earlier in the design process. So initially the design of new drugs are based on the prototypical molecule, usually a natural product, and making structural modification. But for designing of new drugs, the understanding of the etiology of the disease and the structure of the receptor where the ligand or the drug will bind, it is very important. And nowadays there is increasing computational power coupled with applicable software, which has led to more focused approaches to the development of new drugs. The launch of a new drug to market, it is a long-term process that will cost billions of dollars, as I said, and the probability of a failure in drug discovery is really very high. About 90% of the new drugs entering clinical trials, they fail to get the FDA approval and reach the consumer market. So therefore, computer-aided drug discovery, it is a novel tool for drug discovery, getting a lot of attention in the pharma industry these days. It has immense potential and promise in the drug discovery workflow. This tool not only reduces the costs associated with new drug discovery by ensuring the best possible lead compounds for the animal studies, but it may also reduce the time to reach the drug to market as well. So it is helpful to identify lead drug compounds. It can predict any pharmacological actions, possible side effects, and also help in improving the bioavailability of the drug. So CADD have the three major roles in drug discovery campaign, you can say. First is, from a large data set, it will reduce to a small data set. That means a large number of compounds into a smaller set of active compounds, which can be further experimentally tested. Then the optimization process, whatever the smaller subset you have selected, out of that you have to check the absorption, distribution, metabolism, excretion together. It is called as the ADMET properties, what it will do inside the drug, that is pharmacokinetic properties will be there. And the third, what is this? It can design new molecules, that is novel molecules it can design. So it can piece together some fragments into a novel chemotype, like okay fine, this is a part where a molecule should bind, and this is another part where the molecule should bind. So what if I piece these two together and it can make a proper molecule to bind to the both of these sites, right? Now how is it done? If we look at the outline of drug design, general principle for drug design through CADD, now you will have the binding site identification. This is the first site in structure based design. It relies on identification of concave surfaces on the protein that can accommodate the drug sized molecules that also possess appropriate hot spots. So what is the hot spot? Let's say any hydrophobic surface or any hydrogen binding site. Okay, basically anything that can drive the ligand binding. Next is the virtual screening. Now virtual screening is the first method of new ligand identification of new ligands for a given receptor. So for that you have to search a number of databases, right, of 3D structures of smaller molecules to find those which will fit the binding pocket of that receptor, the one which you have already identified in the first step. So that will, you can use some docking programs for that, approximate docking programs. It will make it much faster. And then you can also do structure activity relationship analysis, like based on the structure what kind of function these two molecules interaction might give, that also you can identify. The next is de novo. De novo structure design of new ligands. It can be accomplished by either a ligand linking or a ligand growing approach. So ligand growing approach means what? A fragment will be docked into the binding site and the ligand will be further extended. How you will extend? Growing, simple terms, growing. So you have to add some functional groups to the fragment. So in these methods, the ligand molecules are built up within the constraints of the binding pocket by assembling small pieces in a very stepwise manner. Now these pieces can be either individual molecules or molecular fragments. The key advantage of such a method is that novel structures can be suggested. Okay. The next is optimization. Optimization, you can do that by evaluating the proposed analogs within the binding cavities. What other possibility can be there if we can make it much better? So out of many analogs, then you can select, okay, my analog, nth analog will have better chance of binding, right? And I already told you, out of that hit to lead, you have already done it before the virtual screening, right? And then next you will test for ADME toxicity prediction, what it will, the pharmacokinetic properties, what it will do to the body, what the body will do to the drug, how it will be taken out of the body, any side effects, how will it be metabolized, how it will be absorbed, or how it will be distributed. All these properties you have to take care of. And the last is docking and scoring. So yes, docking, it means to find the best matching between the two molecules. It's like lock and key. It includes finding the right key for the lock. Now to place a ligand, a small molecule that is, into the binding site of a receptor in the manners appropriate for optimal interactions with the receptor, that's what docking will be meant for. And it is also to evaluate the ligand receptor interactions in a way that may discriminate the experimentally observed mode from others and also estimate the binding affinity, right? Now if you look at the workflow of the methods in CADD, so you have the drug discovery and drug development is later, that is preclinical and clinical. So here itself you can do a lot of CADD associated steps, that is your pharmacophore mapping, all these terms you will understand later. So pharmacophores are certain kind of properties which can be identified in the molecule, like some hydrophobic groups or some charged groups, right? Protein structure prediction, target druggability. Then before you go for lead optimization, yes, you have to dock, you have to test whether they are binding properly or not. You can do virtual screening, so you can find out the most favorable bioactive compounds with the help of information about the protein target or known active ligands, right? Now molecular docking, there is one more which can be done as in silico ADME prediction. So molecular docking is in silico method, means through computers. It predicts the placement of small molecules or ligands within the active site of the target protein. Now it is mainly used to accurate the estimation of the most favorable binding sites and also the bioaffinities of ligands with the receptor. Presently it has been broadly applied even to virtual screening for the optimization of the lead compounds. Okay, so then you have the target, this is the flow chart: target protein selection, sequence alignment, homology modeling, molecular dynamics, protein validation. So yes, if all the steps have been correct, if you have got your appropriate results, then you see if there is the best match whatever you are getting, you will generate the derivatives, or if the protein validation is not done properly, then you go for pharmacophore generation. Okay, and you will predict the activity, compare with some bioassay, and then only you will have your drug candidate. So this is the structure based virtual high throughput screening. It is called as VHTS. Okay, so this structure based virtual high throughput screening selects for ligands predicted to bind a particular binding site, as opposed to the traditional high throughput sequence that experimentally asserts general ability of a ligand to bind, inhibit, or allosterically alter the protein function. So the key steps will be preparation of the target protein, developing the library for docking, and determination of some favorable binding position for each compound, and then positioning of those docked structures. Alright, so as I already said, the traditional application of CADD is in lead discovery and optimization. Today the application extends in the direction of target identification and validation, and even towards the preclinical studies. So through mostly through ADME prediction. Now in the drug discovery and development process, CADD is usually used for three major purposes, which I just told you, right? So it can instruct the optimization of lead compounds, screen them, and also design the new novel compounds. So either by adding or modifying the groups. So whatever's this example of applying CADD in different stages of the drug discovery, that is what is illustrated over here. We start from applying homology modeling to generate a target structure, followed by molecular dynamics simulations, so that we can optimize the target structure. Then the protein structure is ready for docking of compound libraries to identify the potential binders. And once the possible binders are identified, combinatorial chemistry can be used to generate a series of such derivatives. But however, if there is no target available, then you have to perform a QSAR, pharmacophore, that can be generated based on the ligand structure and activity information, where key pharmacophore features can be achieved for searching the same classes of binders to the target. Then further you will have the DMPK properties of the binder, such as the ADMATE. It can also be predicted by CADD tools and used to compare with bioassay data. So if a compound can pass all the steps above, then only it will become a drug candidate for the following clinical trials, right? Now there are two kinds, mainly two types of approaches for drug design through CADD. So one is the structure based drug design, another one is ligand based. Now this is a very direct approach. You have the structure or the target based on which you can build your drug molecule, and this is the indirect approach. Okay, so in LBDD, ligand based drug design, 3D structure of the target protein is not known, but the knowledge of ligands which bind to the desired target, that is known. So these ligands can be used to develop a pharmacophore model or molecule which possess all necessary structural features to be able to bind to a target active site. Generally, ligand based techniques are pharmacophore based approach and quantitative structure activity relationships, that is QSAR. In LBDD, it is assumed that compounds which have similarity in their structure, also having the same biological action and interaction with the target protein, those are identified, right? And structure based, the structure of the target protein is known and interaction for bioaffinity for all tested compounds calculate after the process of docking. So to design a drug molecule, it will show better interaction with the target protein, right? So ligand based, it relies on the drug, on the knowledge of other molecules that will bind to the biological target of interest. So for that you have to derive a pharmacophore model that will define the minimum necessary structural characteristic to the molecule which it must possess in order to be able to bind to the target. Now a model of the biological target, it may be built based on the knowledge of what binds to it, and this model in turn may be used to design new molecular entities that interact with the target, and alternatively a quantitative structure activity relationship QSAR, in which a correlation between the calculated properties of molecules and their experimentally determined biological activity, so that may be derived. So these QSAR relationships in turn may be used to predict the activity of new analogues as well. And yes, structure based, it relies on knowledge of the 3D structure of the biological target, which you can obtain through either X-ray crystallography or NMR, nuclear magnetic resonance spectroscopy. So if an experimental structure of a target is not available, it is possible to create a homology model of the target based on the experimental structure of a related protein. So that is called as homology modeling. So it is also called as comparative modeling of protein, which refers to constructing an atomic resolution model of the target and an experimental 3D structure of a related homologous protein, which will serve as the template. So using the structure of the biological target, candidate proteins that are predicted to bind with high affinity and selectivity to the target, they may be designed using some interactive graphics to test the homology. And it also depends upon the intelligence and knowledge of a medicinal chemist, whether they are able to identify the actual process and the molecule they can identify properly, and definitely the various automated computational procedures which may be used to suggest new drug candidates. So CADD speeds up the process of drug design with the help of computers at different stages. One is hit identification using virtual screening, then hit to lead optimization, which I told you about, the QSAR and structure based design, and the lead optimization, so of other pharmaceutical properties, so that they will be able to maintain the affinity. There are some tools and software for CADD. So you have databases and draw tools, the PDB, then ChemBL, Zinc 15 database, PubChem, Drug Bank. So these all are the databases and the draw tools. Then you have molecular modeling and homology modeling. As I already told you, homology modeling is in which a template and target protein, they will share the same evolutionary origin. So this comparative modeling technique, it will involve the some stages that is to serve as a template for structure identification of related protein, then replica of coordinates for confidently aligned regions, and then arrangement of alignment of both target and template protein, and also build missing atom coordinates of the target structure. Finally, you have to do some evaluation and model refinement. For that also tools are there, modeler is there, right? And for binding site prediction and docking, so for homology modeling you have Swiss model also along with modeler. Binding site prediction you have Cavar, MetaPocket, then MedSumo. Then docking, for docking you will have AutoDock, SwissDock, Docking Server, iGemDock. These are your tools. Ligand design screening for QSAR. So screening you can have Farmer or PharmaGist. Then Ligand Scout, some are paid, some will be freely accessible. Discovery Studio. For QSAR you have ClockP, Avogadro, MolDB, and lastly for the binding of free energy estimation and ADME toxicity. So ADME toxicity you will have Astroplast, Swiss ADME, etc. Right? Now the question is why you must know it, right? So computer-aided drug design, it is an efficient tool in the area of drug discovery and development. Through it, web can find the most promising drug candidate in a very cost-effective way. Now the extensive variety of computational tools used in drug discovery campaigns suggests that there are no fundamentally superior techniques. The performance of methods varies greatly with the target protein, the available data, and even available resources. So CADD has had a significant impact on the discovery of various therapeutics that are currently helping treat the patients. So despite the successes, it also faces challenges such as accurate identification and prediction of ligand binding modes and affinities, right? So it always provides a hope for betterment in drug discovery area. In the past years, through CADD, many impressive researches are achieved. So it will play a very much important role in the future as well. Okay. So the phenomenon of drug polymorphism is one of the challenging areas in drug discovery, and it occurs when a drug has different forms which differ structurally and found chemically identical. So this can have a great implication on the impact on the success of the drug. So field of CADD is continuously evolving with these improvements being made in each and every area. And with the current successes, there is definitely a promising future for computational methods to aid in the discovery of many more therapeutics in the future. Now, unfortunately, diseases are continuously increasing because of lifestyle, diet, whatever you say, and even the atmosphere. So, definitely, drug requirement will always be in the market, and through CADD, many job opportunities will also open. This is a very technologically advanced field, so with current achievement you have a real promising future, so that you can not only help in the mankind but also be in a win-win situation where you also can make money if you are going into this field, isn't it? Now the thing is, there are some related field knowledge enhancements which you can do along with this drug discovery, CADD drug discovery. Okay, so one of the most interesting and challenging field a life science graduate can pursue is in clinical trials. These are all related things. So a career in clinical trial will offer you some job opportunities, good ones in the future. So it is one of the booming industries where recent graduates and experienced candidates can find rewarding careers. Then clinical data management, it is a critical technique in clinical research with the help of which statistically sound, reliable and high quality data it can be generated from various clinical steps, right? And one of the most interesting and challenging field is pharmacovigilance, which is most critical step involved in the drug development and clinical trial process. So pharmaceutical companies, IT industry, CRO, they are continuously on a hunt for such talented and skilled professionals in pharmacovigilance. Biotechnica offers certification courses in all this. So in case you want to expand your domain, you're already in this field or you're interested to change your field, so definitely these can be really good options for you. There are some associated tools and techniques also which you can go for. that is chem informatics and drug discovery, molecular modeling. So what I was saying, it's a collection of computer based techniques for deriving, representing and manipulating the structures, the reactions of molecules and those properties that are dependent on these chemical 3D structures, right? And then is also a course on molecular docking. They are all self-learning courses. They are designed to learn various types of docking. They are used to determine the interaction of two molecules. And vital tool we already saw. It is what it is in CADD, right? Then you have related innovative approaches. The one is your precision medicine. Okay, so in the present day, the complexity of diseases, they are increasing, and there is a need for quick and least invasive treatment. So precision medicine is being extensively studied by the leading pharmaceutical companies and the research institutes also. Okay, precision medicine means tailoring the medical treatment to the individual characteristic of a patient. So if you are in the drug development field, definitely this is a topic which you should be aware of. Okay, and everyone after Dr. Jennifer Doudna winning the Nobel Prize and Emmanuel Charpentier, so everyone is talking about CRISPR now. So any kind of novel research methodology in drug discovery could save many lives and money. So CRISPR-Cas9, the recently harnessed genome editing technology, it is quite a candidate in accelerating progress through all these steps. So this course will help you to develop their knowledge and understanding of how drugs and medicines are discovered and the steps and also the technology which is involved in this, right? Some interdisciplinary topics are also there. For example, artificial intelligence. Definitely the goal is to enhance the knowledge of students with the help of some AI technologies. So any enthusiastic participants, they can join and learn how AI can be used in the biotech and the healthcare industry. Then the concept of MATLAB, it is a programming platform developed for engineers and scientists. Using this one can analyze the scientific data to a greater extent. Okay, and MATLAB has become a tool for choice for high productive research development, and definitely you can go for this one as well. Then bioinformatics, I need not tell you, it is a blend of multiple areas of study including biology, data science, mathematics and computer science. So the field focuses on extracting new information and massive quantities of biological data and requires that scientists know the tools and methods for capturing, processing and analyzing large data sets. So definitely in drug you have to play with large data sets, and this can really help you enhance your knowledge. There are other beneficial learning of related fields, for example, forensics and pharma, intelligence, metabolomics and synthetic biology. Synthetic biology, it's an emerging field. It involves the design and construction of new biological entities in case you want to create in the field of drugs also. Metabolomics you will require because it deals with large scale study of metabolites found within the body cells and tissues or organisms. And forensics, definitely the pharma intelligence, it's also an associated course. And it is important to stay updated about most used techniques worldwide. That is, these are the prerequisites for any kind of processes: molecular techniques, then spectroscopic techniques, PCR, right? So these are fundamental tools in physics, chemistry and indispensable technique if you are going for biomedical research, right? So promising fields of research and technology, you have vaccine design, you have nanobiotechnology, then stem cell technology, you have cancer biology, also virology, because drug means it will include all this various areas, isn't it? And finally you have some additional skill enhancements. For example, design of experiment. It's important statistical tools for data collection and analysis used in a variety of experiments. And this will be of everyone's interest, that is research methodology and scientific writing course. It is very important to know the methodology of planning your research from the beginning. So definitely these courses, certified courses, self-timed ones, are really a great addition to your CV and gives a huge weightage. That's all from my end. Thank you everyone for your time and attention. Over to Dr. Tanushri.
Thank you, Dr. Mimala ma'am, for enlightening us about CAD and how it is done. Hello everyone. I'm Dr. Tanushree and I welcome you all to second session of this webinar on computer-aided drug designing. As we all know, coronavirus has become a major public health concern. Since the severe acute respiratory syndrome coronavirus outbreak occurred in year 2002. The continuous evolution of coronaviruses was further highlighted with the emergence of Middle East Respiratory Syndrome coronavirus outbreak in year 2012. Then swine acute diarrhea syndrome coronavirus outbreak in year 2017. And then in late December 2019, the world woke to a reality of a pandemic of coronavirus disease, COVID-19. COVID-19 elicited by severe acute respiratory syndrome coronavirus 2. The potential to cause life-threatening respiratory failure and rapid transmission puts COVID-19 in the list of public health emergency of international concern. In last 2 decades, this is the third deadliest coronavirus pandemic following SARS and Middle East Respiratory Syndrome. Although the fatality rate of SARS-CoV-2 is currently lower than SARS-CoV, but the virus seems to be highly contagious based on number of cases to date. The onset of new diseases, deaths of millions of people due to life-threatening diseases all around the world demands rapid development of promising drugs in the treatment of disease and in maintaining human health. The global corona pandemic has realized the importance and need for drug discovery and development. So when we talk about drug discovery and development process, we know that it is the process of bringing a novel drug from bench to bedside. Drug discovery and development process is a very complex and time-consuming process because if you look at the drug discovery process, it involves several steps. There are several core steps that are carried out during drug discovery. And the very first step is basic research where one needs to look for the disease for which one needs to develop a drug. And then drug discovery process which starts with identifying a potential druggable target for a specific disease, and then work to discover and optimize drug compounds that can elicit an effect on a specific biological target implicated in a disease in the hope of treating it. Then several tests are also performed like preclinical testing, which is designed to deliver important information about a drug candidate, efficacy and safety. That is to deliver information about a compound which has a potential to develop into a drug regarding its efficacy, its effectiveness, its safety before it can be used in human subjects. So numerous questions are addressed at this particular stage, like what does the drug do to the body or what does the body do to the drug? Is the drug safe or not? And then clinical trials are also designed to answer specific research questions related to an investigational new drug. Because unless you actually do human volunteer trials, validated compounds, the compound which have been approved or which have passed the preclinical tests, they will not become a drug or cannot be introduced into the market if they are not approved through or they pass through the clinical trials. So as you can see here, the researcher starts with thousands of compounds, but the success rate is very low. Only 1 or 2 compounds, they actually receive approval from relevant regulatory authority. So we can say that bringing a new drug to the market is a costly process in terms of money, in terms of manpower, and in terms of time. Drug discovery and development, it takes an average of 10 to 15 years. with an approximate cost of US dollar 800 million to US dollar 1.8 billion. So in this current scenario of global health emergency, which demands drugs to enter the market faster while passing all the trials and all the tests. So how can these drugs can enter into the market faster? So the one way to make this possible is by advent of computational tools and methods in drug discovery process. Nowadays, computer is used in almost every field. So question arises, can a computer help us in bringing the drugs, helping us to enter the drugs into the market faster? Or in current scenario, can a computer help us finding a treatment, rapid treatment for COVID-19? Now, when we talk about computers in drug discovery process, that doesn't mean we can replace a medicinal chemist or a synthetic organic chemist or a physical chemist in helping in designing new drugs. Computer aided drug designing. Computers are basically acting as an aiding tools. They are used as aiding tools and now widely used as a new drug design approach. Computer aided drug designing, it is also known as in silico screening, and it has become a powerful technique. It has become an effective and indispensable tool in the long process of drug discovery and development. It is utilized in various phases of drug discovery and development process because of its various advanced features. The advanced features of computer-aided drug designing accelerates the conventional drug discovery process. So what are these features? If we look at the drug discovery process, we know that very first step is that we need to identify a particular target. Now this target, it can be a protein, it can be enzyme, it can be nucleic acid. So not only target identification, but we need to validate it. Which means that we need to use the tools to be sure that this is only the target on which the drug is acting. Then we need to identify a candidate as well. So generally any compound which is approved by regulatory bodies, before they are introduced into the market, they are not called as drug. They are called as lead compounds. Compound which has a very good activity, or you can say a compound which look very promising, which has the potential to develop into a drug. So it is called as process of identifying such compounds and optimizing. So it is the process of lead optimization. So we optimize, once we identify the promising compounds, we optimize them, because the compounds which actually acts very well in laboratory condition does not mean that it has all the properties of a drug. And when we talk about the properties of the drug here, we are referring to the drug likeness property. And what are these properties? Absorption, distribution, metabolism, and excretion. Because we know that drug is something that is taken inside the human body. So it should not be toxic, it should not cause any side effects. It should have a good absorption. So these are drug likeness property, and we also have to modify these properties so that the lead compound does not have only the good activity but also good drug likeness property, which is called as absorption, distribution, metabolism, and excretion. So in this drug discovery process, whether it's target identification, hit identification, lead optimization, and preclinical trials, and all these four processes, computer-aided drug designing plays a very, very important role. So what are the various approaches which are applied in computer-aided drug designing? So various approaches which are applied are the tools such as combinatorial library generation, then molecular docking, virtual high throughput screening, then quantitative structure, activity relationship, comparative modeling, absorption, distribution, metabolism, excretion, toxicity studies. So these have become integral part of modern drug discovery process. Different approaches, stages of designing, docking, pharmacophore modeling, homology modeling, these are all backbone of computer-aided drug designing process. And if we talk about the current scenario of the drug discovery process, it involves several disciplines like chemical and structure biology, computational chemistry. So computational chemistry in order to understand the three-dimensional aspects of the drug receptor interaction, and using technologies like automation in which high throughput screening, one can screen lot many compounds. And this screening helps us in filtering the compounds which do not have the potential to develop into a drug, or unwanted compounds can be screened, can be filtered. So this screening helps the drug discovery process, leads to drug discovery process faster, where and one can synthesize millions of compounds as soon as possible. So if we compare the conventional and modern drug discovery approach, we will find that the conventional approach is completely based on trial and error method. Like a researcher start with thousands of compounds, 30,000 to 50,000 compounds, and testing each compound, synthesizing them and testing them in the wet lab. So just imagine you're working on so many compounds, and we know that it's not that all 50,000 will have the potential to develop into a drug. So it's a very costly, it's a very time consuming process. So the basis is blind screening. You're simply performing testing all the 50,000s of compounds. However, if you compare it with modern approach where computer-aided drug designing is used, it's more logical. You know, need not to work on all the, like you need not to carry all the 50,000 compounds in the wet lab. You filter, you screen these 50,000 compounds using computational tools, and out of these compounds, whichever you find suitable, like let's say 20 or 30 compounds you find suitable, so then you can take these compounds to the wet lab and you can further test them. So just imagine the cost, the resources that are utilized in testing 50,000 compounds and testing and for testing 20 to 30 compounds. So there's a big difference. So that is why modern approach, the drug development cost has reduced about one third. Use of CADD approaches has reduced the cost of drug discovery and development up to 50%. And also time. The drug development time has also significantly reduced. Then it does not involve a sequential process. Parallel steps are involved. Like it's not that once you, like after completing the one step, then only you will move on to the next step. So it's not sequential, it's parallel. You can study target structure at a time. You can study target structure as well as you can look for lead compound. You can identify that. You can perform docking. You can study about quantitative structure activity relationship. So modern approach is very specific and also target based. So whether we talk about time or cost or accuracy or the ability of a process to provide information about the disease or reduce screening, less manpower, these are all the advantages which are offered by computer-aided drug designing in modern drug discovery approach. And this is the reason that employment of computer-aided drug discovery techniques has become essential in top pharmaceutical companies and research groups. Specifically, the use of CADD in preliminary stage of drug discovery. to expedite the drug development process in a more cost efficient way and to minimize failure in the final stage. Now coming to computer aided drug designing applications. Computer aided drug designing offers a lot of application, and the very first application is analysis of target structure. So one can predict the target structure. Now this target can be enzyme, it can be a protein. So structure, why it is important? Because we have to look for binding site where the drug can bind. So prediction of protein structure, because most of the targets are the enzyme or the protein. So prediction of protein structure, it can be done by various methods like homology modeling, and also one can perform docking to detect the target for detection of the target site. Then candidate molecule generation. So as I told you in the previous slide as well, that one need to identify the compound which has a potential to develop into a drug, and we need not only to identify but we also have to optimize it. So optimizing for various properties, various physicochemical properties, and then multi-target drug searching and designing. So even computer-aided drug designing techniques are very useful in searching drugs against multiple targets. And this could be performed in which various hits can be generated against multiple targets. Then quantitative structure activity relationship. Now this QSAR, it is based on the concept that if you change the structure of a molecule, then its function also gets changed. This is because we know that function of a molecule depends on its structure. So structure activity relationship. It completely believes that if you change the structure of a molecule, its function also gets changed. So how much the activity is getting changed. So to answer this, quantitative structure activity relationship was developed to provide the information about how much change in activity occurs when you change the structure of a molecule. Then we can also study about drug receptor interaction, that how the drug they interact with the receptor, like whether with high specificity, with high affinity. So we have to look for good interactions, promising interactions. It should not be that these interactions are very loose and simply the drug can, in different conditions, it can simply remove from the target. So this interaction has to be very much specific, need to be very much, the interaction should occur with high affinity. So we can perform docking of protein-protein or protein ligand. So several types of dockings are there. So docking is a method where you study, where we want to see the interaction between the drug and the receptor. Then we can eliminate compounds of undesirable properties. So just as I told you that here we used in vitro in silico screening method. CADD it uses in silico screening method. So using that we can filter the compounds with undesirable property. You need not to carry all the all the compounds up to the end of the experiment. Simply you first initially filter the compounds, like from thousands of compounds you can filter 20 or 30 compounds which you think that they are very much promising and they have the potential to develop into a drug. So that those 20 to 30 compounds you can carry, you can take to the wet lab and can perform further experiments. Then absorption, distribution, metabolism, excretion, and toxicity prediction. This is very, very important because drug is something that you are taking from outside to inside the body. So this drug should not cause any toxic effect, should not cause any side effect in your body. So we can study the lead compounds, we can study their pharmacokinetic properties like ADME, that is A for absorption, D refers to distribution, M refers to metabolism, E refers to excretion, and as well as you can get information or get knowledge about the toxicity. Then development of potential therapeutics. So studying QSAR and studying about the target structure and identifying and optimizing the lead compound, we can develop the potential therapeutics which can help in treating lot many diseases, whether it can be a cancer or treatment of the diseases caused by viruses and many more. And then coming to drug repurposing. This is a very, very important application of computer-aided drug designing. So when we talk about drug repurposing, we here it refers to the process of identifying new therapeutic use for old, existing, or available drug. So we can say that it is an effective strategy in discovering or developing drug molecules with new pharmacological or therapeutic indications. So using drug repurposing, we can develop rapid treatment for the diseases caused by viruses, and that means we can develop the treatment for COVID-19. So several studies were conducted and it was observed that novel potential inhibitors against SARS-CoV-2, they were identified using virtual screening. So computer aided drug design tool, which is like virtual screening, which is a computer aided drug designing tool. It was used to screen a database of 44 million compounds to find the compounds that have the potential to inhibit the surface of glycoprotein, which are responsible for virus entry and binding. So compounds with promising physicochemical properties and favorable molecular interactions with target protein can be selected, and these selected compounds they can further undergo lead optimization so that they can further developed as drug and can be used in treating COVID-19 diseases. So ADME and physicochemical properties, they were predicted for the selected compounds. So if you compare to the experimental means, computer-aided drug designing based approaches, they are more efficient in providing possible treatment for solutions for epidemic disease outbreaks like COVID-19. So we can say that it is very efficient to apply the computer-aided drug design technique to quickly identify promising drug repurposing candidates. So one such drug is remdesivir. So it is a nucleoside analog which was originally developed against the Ebola virus. Now, on the basis of current knowledge about the use of this drug in SARS-CoV-2 infection, it is considered as a potential drug candidate for repurposing against the COVID-19. And that is the reason, like, that is why we need not to wait for even 6 to 7 years as well. Using drug repurposing, we can use the old drugs, we can use them for the new targets, that is new viruses, new viral structures that were being identified. And that is why most of the research is based on drug repurposing for rapid, for looking for rapid treatment for COVID-19 diseases. And many more drugs are like this is only one example. Many more drugs, many researches are performed where different drugs are tested. They are drug repurposing is performed so that we can find a treatment for COVID-19 diseases. So here are some drugs which are developed using computer-aided drug designing. So drugs which are developed using CADD, these are effective against several viruses, like some are hepatitis C virus inhibitor, some are active against influenza A and B virus. Then antihypertensive drugs are developed. The drugs for rheumatoid arthritis, then drugs for liver cancer, and also the inhibitor of HIV virus. Such drugs are developed. So as I told you about drug repurposing, so we can say that yes, CAD is to the rescue for COVID-19. Using computer-aided drug designing tools, we can develop the rapid treatment for COVID-19 diseases. So computer-aided drug designing, it's a very fast and reliable tool which is of essence in current scenario. So as I told you that computer-aided drug designing is now become an indispensable tool, a very powerful tool in new drug discovery process. So we can expect a global rise from year 2018 to 2026. A global computer-aided drug discovery market insight is shown over here, and this is because of the increase in research in oncology segment, in cardiovascular disease segment, research institute and pharmaceutical company. This is because we know that with the change in environmental conditions, with increase in population, and with the change in lifestyle of the people, we hear about new diseases every day. And this is the reason that the like whether we talk about cancer, so we know that the cancer cases has been significantly increased in recent years, and this is because of the change in environmental conditions and the change in lifestyle of the people, and even the heart diseases has also increased. So a lot of research which uses the computer aided drug designing tools in order to find the treatment, the rapid treatment, development of rapid drugs for treatment of such diseases like cancer and heart diseases. And this is the reason that because of growing healthcare expenditure across the globe, which uh, we can anticipate, like this can anticipate to drive the computer aided drug discovery market growth. So we can say that the future in drug discovery is all based on computer aided drug discovery process. The updation, the development of new tools which can be used in computer aided drug designing so that we can make the drug discovery process much more faster and much more effective. So question arises, if this computer aided drug designing is such a promising and such a important technique, so where to find the latest information about it? Where to find, where to know about the basics, where to know about the updates related to computer aided drug designing? So here is the answer. Biotechnica presents the computer aided drug designing certification course where you can get not only the basic information about computer aided drug designing but also the latest information. So what are the modules of the certification course? So basically this certification course has 5 modules, and module 1 is basically comprises of 6 sessions. So this module 1 is all about introduction to computer aided drug designing. That is, it is all about basics. So here in this session, here in this module, you will get to know about the overview of process of drug discovery. You will get to know about the basics of you will get to know about bioinformatics, what is proteomics, what is genomics, what are different databases that are used, and what are different sequence search tools that are used in bioinformatics, and specifically BLAST that will be discussed in detail, and then factors affecting the drug discovery and what are the different types of computer aided drug designing. So the module 1 is all about basics, and as I told you that computer aided drug designing, it involves a lot of approaches. Like one need to be aware of different databases, one need to be aware of different search tools, if you need to know about, if you need to use the computational tools in new drug discovery process. Then module 2 is about ligand-based drug design, or we can say pharmacophore design. So this module is all about how the drugs can be designed using ligand-based approach. And here in this particular module, the pharmacophore modeling, QSAR analysis, then similarity search and R group decomposition, matched pair analysis. These are the topics that will be discussed in detail. And not only they will be you will not know only about the basics, but also through several and also through demo videos you will get to know, you will better understand. So the demo videos are also there. You will better understand about what is QSAR, what is how R group decomposition is done. Then module 3, it is about structure-based drug design, and it includes 7 sessions. So here in this module, one will know about the basics of docking, how to perform docking analysis, what is data mining, and in case of non-availability of target structure, which approach to use, like how to perform homology modeling, how to detect, how to identify the active site, and what are the different approaches that are used in structure-based drug design. So all these topics are discussed in detail in this module 3. Then module 4 is about significance and limitations. Significance and limitations of computer-aided drug designing in drug discovery and development. Now this is again very important topic that one need to know, because we know that there is no technique which like every technique has certain advantages and disadvantages. So unless and until you are not aware about the limitations, you cannot work on finding the solutions or how to overcome these limitations. So that is why the knowledge about the significance and limitations of computer-aided drug designing, one need to know. It is very important in drug discovery and development process. And then module 5 is about case studies. So if you see the module, it is designed in such a way that if you study this course, you will get to know about lot many things. Like you are getting the information, you are knowing about bioinformatics, the databases that I used, the search tools that I used, and also basically you are studying about the omics, whether it's the proteomics or genomics, because you know we are living in the omic era, and one need to know about these, how these proteomics and genomics plays a very important role. Then several databases, like when we are talking about computational tools, so definitely databases has a lot of role to play over there, and one need to know that what are the different protein databases, nucleic acid databases, and how one can use them in computer aided drug designing process, or computer aided drug designing. uh, and then also several computational tools which involves the machine learning, artificial intelligence, deep learning, and the basic drug discovery process, and also what are the significance and limitations of computer aided drug designing in drug discovery process. All that one will know in a single certification course. We all know that computational tools are used in almost every field nowadays. So it's not that if you want to go into drug designing, then only this course will be beneficial. Even you are from biotechnology or from pharmacy or nanotechnology or agriculture or molecular biology field, then also this computer aided drug designing certification course has a lot of value, because you will be able to know about lot many things in a single certification course. So question arises, what are the benefits of computer aided drug designing in career? And computer aided drug designing provides a lot of advantage in one's career. That is, you can work as a drug design research scientist, you can work as research assistant or associate, and then also if you have interest in academics, you can go for professorship, and then computational biophysicist, computational chemist, you can work if you have the background, or if you have the knowledge about, or if you have done course on computer aided drug designing, then you can work as CAD technician, and also you can go for your own business development. So CAD certification course, it offers several benefits. So very first benefit is that you will learn new skill. So no matter where you are in your current career, it's never too late to learn and develop new skills. Learning new skills keeps our mind engaged and body active, and it helps us get new and knowledge-based perspectives on the world around us. And we all know that learning is always exciting and rewarding. Then it helps achieve your goal. When you learn a new skill, it definitely opens door for you to work in different type of companies, in different type of research institutes where they have the similar requirement, where they need a candidate having the background of with the knowledge of bioinformatics or with the knowledge of computational tools. Then you will learn more in a short period. So as I told you that there are 5 modules only in this course, and you will learn a lot about the basics and also the recent information. So you will be updated with the latest knowledge, and learning a new skill also makes a transition into professional career easier, and also makes your CV stand out of the crowd. And also, as you know, that it's certification course. So we all know that certificate is always motivating, and the new skills always helps you stay competitive. So these are the several benefits of this computer-aided drug designing certification course. So with this, I have reached to the end of this session, and I would like to end this session with 2 beautiful quotes. The more that you read, the more things you will know. The more that you learn, the more places you will go. So as I told you that there are several benefits of learning new skills. So as in this course, you will know about lot many things. So as you know about lot many things, you will learn about them. And when you learn about new things, definitely there will be lot many options available to you, and you will be able to look the things with different perspectives. And also, no matter how much experience you have, there's always something new you can learn and room for improvement. Thank you.
The global corona pandemic has realized the importance and needs for drug discovery and development. The onset of new diseases, the death of millions of people due to life-threatening diseases all around the world demands rapid development of promising drugs in the treatment of disease and maintaining human health. Drug discovery is a multi-step and interdisciplinary process. The research and development of drugs is a time-consuming and expensive process that can take approximately 10 to 12 years. But the current scenario demands drugs to enter the market faster while passing all the trials and tests. One way to make this possible is by advent of computational tools and methods in the drug discovery process. Computer-aided drug design, CADD, also known as in silico screening, has become a powerful technique because of its utility in various phases of drug discovery and development through various advanced features. It can be combined with wet lab techniques to elucidate the mechanism of drug resistance, to search for new drug targets, and to design novel drug candidates for both known and new targets, compounds, screening, and optimization. In recent years, CADD has now become an indispensable tool in the long process of drug discovery and development. Thus, Biotechnica presents an online certification course on CADD, Computer-Aided Drug Designing. The course will cover basics as well as detailed information about CADD, ligand and structure-based drug design, significance as well as limitations of computer-aided drug design. So who is this course for? Students pursuing BSc, BTech, MSc, MTech, PhD, postdocs in any science field, B Pharma, M Pharma, who aspire to work on cutting-edge research technologies should enroll. Candidates interested in pursuing intensive research in one or more of the following domains can also enroll: biological science, computer science, pharmacy, agriculture, biotechnology, chemistry, molecular biology, medicine and clinical research will definitely benefit from the course. Once you become a specialist in CADD, an array of opportunities open up. One can work as drug design research scientist, CADD investigator, computational chemist, molecular modeler, or principal investigator. These are just a few to list here. At the end of this course, there is a knowledge test based on modules and sessions discussed in the course. Just pass the test and you get certificate in CADD. Enroll in Biotecnica's CADD certification course to master one of the trending tools in the industry. Get certified now and get future ready.